Welcome to Biology Masterclass. In this masterclass today, we will learn 200 short questions and answers on cell biology and genetics. Are you ready? Let's begin. Cell biology. Question 1. Did you know? What is the basic unit of life? The cell is the basic structural and functional unit of all living organisms. Question 2. Did you know? Who discovered the cell? Robert Hooke discovered the cell in 1665 while examining cork tissue under a microscope. Question 3. Did you know? What are prokaryotic cells? Prokaryotic cells are simple cells without a membrane-bound nucleus, found in bacteria and archaea. Question 4. Did you know? What are eukaryotic cells? Eukaryotic cells are complex cells with a membrane-bound nucleus and organelles, found in animals, plants, fungi, and protists. Question 5. Did you know? What is the function of mitochondria? Mitochondria are the powerhouse of the cell, producing ATP through cellular respiration. Question 6. Did you know? What is the function of chloroplasts? Chloroplasts conduct photosynthesis, converting light energy into chemical energy stored in glucose. Question 7. Did you know? What is the cell membrane made of? The cell membrane is composed of a phospholipid bilayer with embedded proteins and cholesterol. Question 8. Did you know? What is cytoplasm? Cytoplasm is the gel-like substance inside the cell that contains organelles and where cellular processes occur. Question 9. Did you know? What is the function of the nucleus? The nucleus stores genetic material, DNA, and controls cellular activities, including growth and reproduction. Question 10. Did you know? What are ribosomes? Ribosomes are cellular structures that synthesize proteins by translating messenger RNA. Question 11. Did you know? What is the endoplasmic reticulum? The endoplasmic reticulum is a network of membranes involved in protein and lipid synthesis. Question 12. Did you know? What is the difference between rough and smooth ER? Rough ER has ribosomes and makes proteins. Smooth ER lacks ribosomes and synthesizes lipids. Question 13. Did you know? What is the Golgi apparatus? The Golgi apparatus modifies, packages, and distributes proteins and lipids to their destinations. Question 14. Did you know? What are lysosomes? Lysosomes are organelles containing digestive enzymes that break down waste materials and cellular debris. Question 15. Did you know? What is the cell wall? The cell wall is a rigid outer layer in plant cells, bacteria, and fungi that provides structural support. Question 16. Did you know? What is the difference between plant and animal cells? Plant cells have cell walls, chloroplasts, and large vacuoles. Animal cells have centrioles and smaller vacuoles. Question 17. Did you know? What is osmosis? Osmosis is the movement of water molecules across a selectively permeable membrane from high to low concentration. Question 18. Did you know? What is diffusion? Diffusion is the movement of molecules from an area of high concentration to low concentration. Question 19. Did you know? What is active transport? Active transport is the movement of molecules against a concentration gradient using energy, ATP. Question 20. Did you know? What is passive transport? Passive transport is the movement of molecules across membranes without energy expenditure. Question 21. Did you know? What is cellular respiration? Cellular respiration is the process of breaking down glucose to produce ATP for cellular energy. Question 22. Did you know? What is photosynthesis? Photosynthesis is the process by which plants convert light energy, carbon dioxide, and water into glucose and oxygen. Question 23. Did you know? What is the formula for photosynthesis? 6CO2 plus 6H2O plus light energy, C6H12O6 plus 6O2. Question 24. Did you know? What is the formula for cellular respiration? C6H12O6 plus 6O2, 6CO2 plus 6H2O plus ATP. Question 25. Did you know? What is ATP? ATP, adenosine triphosphate, is the primary energy currency of cells. Question 26. Did you know? What are vacuoles? Vacuoles are storage organelles that hold water, nutrients, and waste products. Question 27. Did you know? What is cell division? Cell division is the process by which a parent cell divides into two or more daughter cells. Question 28. Did you know? What is mitosis? Mitosis is cell division that produces two identical daughter cells with the same chromosome number. Question 29. Did you know? What are the phases of mitosis? The phases are prophase, metaphase, anaphase, and telophase, followed by cytokinesis. Question 30. Did you know? What is meiosis? Meiosis is cell division that produces four non-identical sex cells with half the chromosome number. Question 31. Did you know? What is the difference between mitosis and meiosis? Mitosis produces identical diploid cells for growth. Meiosis produces diverse haploid gametes for reproduction. Question 32. Did you know? What is a chromosome? A chromosome is a thread-like structure of DNA and proteins that carries genetic information. Question 33. Did you know? What is DNA? DNA, deoxyribonucleic acid, is the hereditary material that contains genetic instructions. Question 34. Did you know? What is RNA? RNA, ribonucleic acid, is a nucleic acid that helps in protein synthesis and gene regulation. Question 35. Did you know? What is the structure of DNA? DNA has a double helix structure with two complementary strands connected by base pairs. Question 36. Did you know? What are the four bases in DNA? Adenine, thymine, guanine, and cytosine. Question 37. Did you know? What are the base pairing rules in DNA? Adenine pairs with thymine, guanine pairs with cytosine. Question 38. Did you know? What is a gene? A gene is a segment of DNA that codes for a specific protein or functional RNA. Question 39. Did you know? What is the cell cycle? The cell cycle is the series of events that cells undergo from formation to division. Question 40. Did you know? What are stem cells? Stem cells are undifferentiated cells capable of developing into various specialized cell types. Question 41. Did you know? What is apoptosis? Apoptosis is programmed cell death, a normal process for removing damaged or unnecessary cells. Question 42. Did you know? What is necrosis? Necrosis is uncontrolled cell death caused by injury or infection. Question 43. Did you know? What are peroxisomes? Peroxisomes are organelles that break down fatty acids and detoxify harmful substances. Question 44. Did you know? What is the cytoskeleton? The cytoskeleton is a network of protein filaments that provides structural support and enables cell movement. Question 45. Did you know? What are centrioles? Centrioles are cylindrical structures that organize microtubules during cell division. Question 46. Did you know? What is the nucleolus? The nucleolus is a structure within the nucleus that produces ribosomal RNA. Question 47. Did you know? What is chromatin? Chromatin is the complex of DNA and proteins that forms chromosomes. Question 48. Did you know? What is a selectively permeable membrane? A selectively permeable membrane allows some substances to pass while blocking others. Question 49. Did you know? What is endocytosis? Endocytosis is the process of taking materials into the cell by engulfing them in vesicles. Question 50. Did you know? What is exocytosis? Exocytosis is the process of expelling materials from the cell in vesicles. Question 51. Did you know? What is phagocytosis? Phagocytosis is the process of engulfing large particles or cells for digestion. Question 52. Did you know? What is pinocytosis? Pinocytosis is the process of taking in small droplets of extracellular fluid. Question 53. Did you know? What is a hypertonic solution? A hypertonic solution has a lower solute concentration than the cell, causing water to enter. Question 54. Did you know? What is a hypertonic solution? A hypertonic solution has a higher solute concentration than the cell, causing water to leave. Question 55. Did you know? What is an isotonic solution? An isotonic solution has the same solute concentration as the cell with no net water movement. Question 56. Did you know? What is plasmolysis? Plasmolysis is the shrinking of plant cell cytoplasm away from the cell wall in hypertonic conditions. Question 57. Did you know? What is turgor pressure? Turgor pressure is the pressure of cell contents against the cell wall in plant cells. Question 58. Did you know? What are enzymes? Enzymes are biological catalysts that speed up chemical reactions without being consumed. Question 59. Did you know? What is the substrate? The substrate is the specific molecule upon which an enzyme acts. Question 60. Did you know? What is the active site? The active site is the region on an enzyme where the substrate binds. Question 61. Did you know? What is enzyme specificity? Enzyme specificity means each enzyme catalyzes only specific reactions with particular substrates. Question 62. Did you know? What factors affect enzyme activity? Temperature, pH, substrate concentration, and enzyme concentration affect enzyme activity. Question 63. Did you know? What is denaturation? Denaturation is the loss of enzyme structure and function due to extreme conditions. Question 64. Did you know? What are cofactors? Cofactors are non-protein molecules required for enzyme activity. Question 65. Did you know? What are coenzymes? Coenzymes are organic cofactors, often derived from vitamins. Question 66. Did you know? What is glycolysis? Glycolysis is the breakdown of glucose into pyruvate, producing ATP and NADH. Question 67. Did you know? Where does glycolysis occur? Glycolysis occurs in the cytoplasm of cells. Question 68. Did you know? What is the Krebs cycle? The Krebs cycle is a series of reactions that oxidize acetyl-CoA, producing ATP, NADH, and FADH2. Question 69. Did you know? Where does the Krebs cycle occur? The Krebs cycle occurs in the mitochondrial matrix. Question 70. Did you know? What is the electron transport chain? The electron transport chain is a series of proteins that transfer electrons to produce ATP. Question 71. Did you know? Where does the electron transport chain occur? The electron transport chain occurs in the inner mitochondrial membrane. Question 72. Did you know? What is oxidative phosphorylation? Oxidative phosphorylation is ATP production using energy from electron transfer in the electron transport chain. Question 73. Did you know? What is fermentation? Fermentation is anaerobic glucose breakdown that produces limited ATP and byproducts like lactic acid or ethanol. Question 74. Did you know? What is lactic acid fermentation? Lactic acid fermentation converts pyruvate to lactic acid, occurring in muscle cells during intense exercise. Question 75. Did you know? What is alcoholic fermentation? Alcoholic fermentation converts pyruvate to ethanol and carbon dioxide, occurring in yeast. Question 76. Did you know? What are the light-dependent reactions? Light-dependent reactions occur in thylakoids using light to produce ATP and NADPH. Question 77. Did you know? What are the light-independent reactions? Light-independent reactions, Calvin cycle, occur in the stroma, using ATP and NADPH to produce glucose. Question 78. Did you know? What is chlorophyll? Chlorophyll is the green pigment in chloroplasts that absorbs light energy for photosynthesis. Question 79. Did you know? What wavelengths does chlorophyll absorb? Chlorophyll primarily absorbs blue and red light, reflecting green light. Question 80. Did you know? What is the stroma? The stroma is the fluid-filled space inside chloroplasts where the Calvin cycle occurs. Question 81. Did you know? What are thylakoids? Thylakoids are membrane-bound compartments in chloroplasts where light-dependent reactions occur. Question 82. Did you know? What is the Calvin cycle? The Calvin cycle fixes carbon dioxide into glucose using ATP and NADPH. Question 83. Did you know? What is carbon fixation? Carbon fixation is the process of converting inorganic carbon dioxide into organic compounds. Question 84. Did you know? What is rubisco? Rubisco is the enzyme that catalyzes the first step of carbon fixation in the Calvin cycle. Question 85. Did you know? What is chemiosmosis? Chemiosmosis is ATP production driven by proton gradient across a membrane. Question 86. Did you know? What is the mitochondrial matrix? The mitochondrial matrix is the innermost compartment of mitochondria where the Krebs cycle occurs. Question 87. Did you know? What are cristae? Cristae are folds of the inner mitochondrial membrane that increase surface area for ATP production. Question 88. Did you know? How many ATP molecules are produced from one glucose molecule? Approximately 30 to 32 ATP molecules are produced from complete glucose oxidation. Question 89. Did you know? What is anaerobic respiration? Anaerobic respiration is cellular respiration without oxygen, producing less ATP than aerobic respiration. Question 90. Did you know? What is aerobic respiration? Aerobic respiration is cellular respiration requiring oxygen, producing maximum ATP. Question 91. Did you know? What is NADH? NADH is an electron carrier that transfers high-energy electrons to the electron transport chain. Question 92. Did you know? What is FADH2? FADH2 is another electron carrier produced during the Krebs cycle. Question 93. Did you know? What is substrate-level phosphorylation? Substrate-level phosphorylation is direct ATP production by transferring phosphate from substrate to ADP. Question 94. Did you know? What is the primary function of the cell membrane? The cell membrane regulates what enters and exits the cell, maintaining homeostasis. Question 95. Did you know? What are integral proteins? Integral proteins are embedded in the cell membrane, often serving as channels or receptors. Question 96. Did you know? What are peripheral proteins? Peripheral proteins are loosely attached to membrane surfaces, often involved in signaling. Question 97. Did you know? What is the fluid mosaic model? The fluid mosaic model describes the cell membrane as a flexible structure with proteins embedded in a lipid bilayer. Question 98. Did you know? What are transport proteins? Transport proteins facilitate movement of specific molecules across cell membranes. Question 99. Did you know? What is facilitated diffusion? Facilitated diffusion is passive transport using carrier or channel proteins. Now let us answer short questions about genetics. Question 100. Did you know? What is the sodium-potassium pump? The sodium-potassium pump actively transports sodium out and potassium into cells, maintaining electrochemical gradients. Question 101. Did you know? What is genetics? Genetics is the study of heredity and variation in living organisms. Question 102. Did you know? Who is the father of genetics? Gregor Mendel is considered the father of genetics for his work on pea plants. Question 103. Did you know? What is heredity? Heredity is the transmission of genetic characteristics from parents to offspring. Question 104. Did you know? What is an allele? An allele is an alternative form of a gene at a specific location on a chromosome. Question 105. Did you know? What is a genotype? A genotype is the genetic makeup of an organism. Question 106. Did you know? What is a phenotype? A phenotype is the observable physical or biochemical characteristics of an organism. Question 107. Did you know? What is a dominant allele? A dominant allele is expressed in the phenotype even when only one copy is present. Question 108. Did you know? What is a recessive allele? A recessive allele is expressed only when two copies are present. Question 109. Did you know? What is homozygous? Homozygous means having two identical alleles for a particular gene. Question 110. Did you know? What is heterozygous? Heterozygous means having two different alleles for a particular gene. Question 111. Did you know? What is Mendel's law of segregation? Each parent passes one allele for each gene randomly to offspring during gamete formation. Question 112. Did you know? What is Mendel's law of independent assortment? Genes for different traits are inherited independently of each other. Question 113. Did you know? What is a Punnett square? A Punnett square is a diagram used to predict offspring genotypes from parental crosses. Question 1 through 14. Did you know? What is a monohybrid cross? A monohybrid cross examines inheritance of one trait. Question 115. Did you know? What is a dihybrid cross? A dihybrid cross examines inheritance of two traits simultaneously. Question 116. Did you know? What is incomplete dominance? Incomplete dominance occurs when neither allele is completely dominant, resulting in a blended phenotype. Question 117. Did you know? What is codominance? Codominance occurs when both alleles are expressed equally in the phenotype. Question 118. Did you know? What is a test cross? A test cross breeds an organism with unknown genotype with a homozygous recessive individual. Question 119. Did you know? What is linkage? Linkage is when genes located close together on the same chromosome are inherited together. Question 120. Did you know? What is crossing over? Crossing over is the exchange of genetic material between homologous chromosomes during meiosis. Question 121. Did you know? What is a mutation? A mutation is a permanent change in DNA sequence. Question 122. Did you know? What is a point mutation? A point mutation is a change in a single nucleotide base in DNA. Question 123. Did you know? What is a frameshift mutation? A frameshift mutation is an insertion or deletion that shifts the reading frame of codons. Question 124. Did you know? What are sex-linked traits? Sex-linked traits are characteristics determined by genes on sex chromosomes. Question 125. Did you know? What are autosomes? Autosomes are non-sex chromosomes. Question 126. Did you know? What determines biological sex in humans? XX chromosomes determine female. XY chromosomes determine male. Question 127. Did you know? What is a karyotype? A karyotype is an organized image of all chromosomes in a cell. Question 128. Did you know? What is non-disjunction? Non-disjunction is the failure of chromosomes to separate properly during cell division. Question 129. Did you know? What is Down syndrome? Down syndrome is a genetic disorder caused by an extra copy of chromosome 21. Question 130. Did you know? What is Turner syndrome? Turner syndrome is a condition where females have only one X chromosome, 45 con X. Question 131. Did you know? What is Klinefelter syndrome? Klinefelter syndrome is a condition where males have an extra X chromosome, 47XXY. Question 132. Did you know? What is DNA replication? DNA replication is the process of copying DNA before cell division. Question 133. Did you know? What enzyme unwinds DNA during replication? DNA helicase unwinds the double helix. Question 134. Did you know? What enzyme synthesizes new DNA strands? DNA polymerase synthesizes new DNA strands. Question 135. Did you know? What is the leading strand? The leading strand is synthesized continuously in the 5 to 3 direction. Question 136. Did you know? What is the lagging strand? The lagging strand is synthesized discontinuously in short segments called Okazaki fragments. Question 137. Did you know? What are Okazaki fragments? Okazaki fragments are short DNA sequences synthesized on the lagging strand. Question 138. Did you know? What is transcription? Transcription is the synthesis of RNA from a DNA template. Question 139. Did you know? What is translation? Translation is the synthesis of proteins from mRNA templates. Question 140. Did you know? What is messenger RNA, mRNA? mRNA carries genetic information from DNA to ribosomes for protein synthesis. Question 141. Did you know? What is transfer RNA, tRNA? tRNA brings amino acids to ribosomes during protein synthesis. Question 142. Did you know? What is ribosomal RNA, rRNA? rRNA is a structural and catalytic component of ribosomes. Question 143. Did you know? What is a codon? A codon is a 3-nucleotide sequence that specifies an amino acid. Question 144. Did you know? What is an anticodon? An anticodon is a 3-nucleotide sequence on tRNA complementary to an mRNA codon. Question 145. Did you know? How many codons code for amino acids? 61 codons code for amino acids. 3 are stop codons. Question 146. Did you know? What is the start codon? AUG is the start codon, coding for methionine. Question 147. Did you know? What are stop codons? UAA, UAG, and UGA are stop codons that terminate translation. Question 148. Did you know? What is the genetic code? The genetic code is the set of rules by which DNA sequences are translated into proteins. Question 149. Did you know? Is the genetic code universal? Yes, the genetic code is nearly universal across all organisms. Question 150. Did you know? What is a promoter? A promoter is a DNA region where RNA polymerase binds to initiate transcription. Question 151. Did you know? What is RNA polymerase? RNA polymerase is the enzyme that synthesizes RNA from DNA templates. Question 152. Did you know? What is a terminator sequence? A terminator sequence signals the end of transcription. Question 153. Did you know? What is RNA processing? RNA processing modifies pre-mRNA into mature mRNA through splicing, capping, and adding a poly-A tail. Question 154. Did you know? What are introns? Introns are non-coding DNA sequences removed during RNA splicing. Question 155. Did you know? What are exons? Exons are coding DNA sequences retained in mature mRNA. Question 156. Did you know? What is alternative splicing? Alternative splicing produces different proteins from the same gene by including different exons. Question 157. Did you know? What is a polygenic trait? A polygenic trait is controlled by multiple genes. Question 158. Did you know? What is pleiotropy? Pleiotropy occurs when one gene affects multiple phenotypic traits. Question 159. Did you know? What is epistasis? Epistasis is when one gene masks or modifies the effect of another gene. Question 160. Did you know? What is genetic recombination? Genetic recombination is the rearrangement of genetic material to produce offspring with combinations different from parents. Question 161. Did you know? What is a gene pool? A gene pool is the total genetic diversity within a population. Question 162. Did you know? What is allele frequency? Allele frequency is the proportion of a specific allele in a population. Question 163. Did you know? What is the Hardy-Weinberg principle? The Hardy-Weinberg principle states that allele frequencies remain constant in a non-evolving population. Question 164. Did you know? What conditions are required for Hardy-Weinberg equilibrium? No mutations, random mating, no gene flow, infinite population size, and no natural selection. Question 165. Did you know? What is genetic drift? Genetic drift is random changes in allele frequencies in small populations. Question 166. Did you know? What is the bottleneck effect? The bottleneck effect is genetic drift caused by a sharp reduction in population size. Question 167. Did you know? What is the founder effect? The founder effect occurs when a new population is established by a small number of individuals. Question 168. Did you know? What is gene flow? Gene flow is the transfer of alleles between populations through migration. Question 169. Did you know? What is natural selection? Natural selection is the differential survival and reproduction of individuals due to phenotypic differences. Question 170. Did you know? What is artificial selection? Artificial selection is selective breeding by humans to produce desired traits. Question 171. Did you know? What is directional selection? Directional selection favors one extreme phenotype over others. Question 172. Did you know? What is stabilizing selection? Stabilizing selection favors intermediate phenotypes over extremes. Question 173. Did you know? What is disruptive selection? Disruptive selection favors extreme phenotypes over intermediate ones. Question 174. Did you know? What is sexual selection? Sexual selection is selection based on mating success rather than survival. Question 175. Did you know? What is fitness in evolutionary terms? Fitness is the reproductive success of an organism. Question 176. Did you know? What is adaptation? Adaptation is an inherited characteristic that increases survival and reproduction. Question 177. Did you know? What is speciation? Speciation is the formation of new and distinct species in evolution. Question 178. Did you know? What is allopatric speciation? Allopatric speciation occurs when populations are geographically isolated. Question 179. Did you know? What is sympatric speciation? Sympatric speciation occurs within the same geographic area without physical barriers. Question 180. Did you know? What is reproductive isolation? Reproductive isolation prevents gene flow between populations, leading to speciation. Question 181. Did you know? What is a homologous structure? Homologous structures have similar anatomy due to common ancestry, but may serve different functions. Question 182. Did you know? What is an analogous structure? Analogous structures have similar functions, but evolved independently without common ancestry. Question 183. Did you know? What is vestigial structure? Vestigial structures are remnants of organs or structures that had functions in ancestral species. Question 184. Did you know? What is convergent evolution? Convergent evolution produces similar traits in unrelated species due to similar environmental pressures. Question 185. Did you know? What is divergent evolution? Divergent evolution occurs when related species develop different traits due to different environments. Question 186. Did you know? What is coevolution? Coevolution is reciprocal evolutionary change in interacting species. Question 187. Did you know? What is molecular clock? A molecular clock estimates evolutionary time based on the rate of genetic mutations. Question 188. Did you know? What is a phylogenetic tree? A phylogenetic tree is a diagram showing evolutionary relationships among species. Question 189. Did you know? What is cladistics? Cladistics is a method of classification based on common ancestry. Question 190. Did you know? What is a clade? A clade is a group of organisms consisting of a common ancestor and all descendants. Question 191. Did you know? What is horizontal gene transfer? Horizontal gene transfer is the movement of genetic material between organisms other than by reproduction. Question 192. Did you know? What is polyploidy? Polyploidy is having more than 2 complete sets of chromosomes. Question 193. Did you know? What is aneuploidy? Aneuploidy is an abnormal number of chromosomes. Question 194. Did you know? What is a genetic marker? A genetic marker is a DNA sequence with a known location used to identify genes or traits. Question 195. Did you know? What is PCR? PCR, polymerase chain reaction, is a technique to amplify specific DNA sequences. Question 196. Did you know? What is gel electrophoresis? Gel electrophoresis separates DNA fragments by size using an electric field. Question 197. Did you know? What is DNA sequencing? DNA sequencing determines the exact order of nucleotides in DNA. Question 198. Did you know? What is CRISPR? CRISPR is a gene editing technology that allows precise modifications to DNA. Question 199. Did you know? What is cloning? Cloning is creating genetically identical copies of organisms or DNA. Question 200. Did you know? What is the Human Genome Project? the Human Genome Project mapped all human genes, completed in 2003.